Namaskar everyone. Welcome to the educational video lecture series by MedDwise Circle. In this video lecture series, we are discussing the fundamentals of clinical chemistry tests which are performed in pathology laboratories. This video lecture series is made in collaboration with Anamol Laboratories Private Limited. In this video lecture series, we are discussing what kind of diagnostic tests are performed in pathology lab specifically for clinical chemistry segment. So without wasting any time, let us go to our lecture series. Please allow me to share my screen. Friends, I hope my screen is visible to everyone. So in this video lecture series, as I discussed, we are only discussing the fundamentals of clinical chemistry test in pathology laboratory. In our previous video lectures, we have already prepared lot of videos on the basics of photometry. In this, we covered topics such as what is wavelength, what is complementary color, what is Beer-Lambert law, and how this is implemented in pathology laboratory. What are the components of colorimeter and what is the difference between colorimeter and spectrophotometer? This was our module number 1. In this module, which is our module number 2, we are discussing the role of enzymes in diagnostic test. Why enzymes are so important? In our previous video lecture, we covered what is the role of enzyme in human body and in diagnostic test. In today's video lecture, we are going to discuss what are the factors that affect the enzyme activity. So without wasting any time, let us start. So before we start the factors which affect the enzyme activity, let us quickly recap in 1 minute what we discussed in our previous video lecture. In previous video lecture, we discussed that enzymes are nothing but they are catalysts that speed up the reaction. The way enzyme function is that enzyme acts on a particular substrate and it forms a substrate and enzyme complex. And this enzyme substrate complex then finally dissociates back where the substrate is converted into the product. Enzymes are used in diagnostic test because enzymes act on one particular specific substrate only. Now here we have seen that enzyme does not take part in the reaction by themselves. They only increase the speed of the reaction, which means that the quantity of enzyme is constant. It does not increase or decrease over the reaction. It is important to understand this concept because in many diseases, the quantity of enzyme does not increase or decrease. Diseases are caused in human body because the activity of enzyme, either it increases or it decreases. With this understanding, we came to also understand what are the attributes of enzyme activity. We have seen that it is important to measure the activity of enzyme rather than the quantity of enzyme. Now, activity of enzyme is dependent on certain attributes. For example, enzyme activity is always specific to one particular substrate, which means that glucose oxidase enzyme will only act on glucose substrate and it will not act on any other sugar. This is the reason why lot of enzymes are also added in the biochemical formulations. We have also seen that enzyme activity enzyme works by way of reducing the activation energy to start the reaction, which means that every reaction requires a minimum amount of energy to initiate the reaction. So the way enzyme functions is to lower the activation energy. We have also seen that enzyme activity can be measured by how much substrate is consumed over per minute or how much product is formed per minute. This is the way we measure the activity of enzyme. We have also seen that the activity of enzyme depends on the environment in which the enzyme is functioning. Now, in this video lecture, we are going to discuss more details about what are the factors or what are the environmental factors that affect the enzyme activity. Enzyme activity in scientific language is more commonly termed as enzyme kinetics. But largely, these two terminologies are interchangeable. So enzyme activity is also referred to as enzyme kinetics. So what are the factors that affect the enzyme kinetics? Largely, we have seen that in pathology laboratory conditions, there are five factors that affect the enzyme kinetics. These five factors are: what is the pH of the reaction, or in simple language, what is the pH in which the enzyme is functioning? Secondly, what is the ionic strength of buffer in which the enzyme is functioning? Third, what is the reagent to sample ratio that is being maintained? Fourth, what is the timing of the or measuring the absorbance? And fifth, what is the temperature at which the enzyme is functioning? Let us try to understand each one of them one by one. First, let's try to understand how pH affects the enzyme activity. Friends, we all know enzyme is protein in nature. So enzyme molecule has many ionic groups. These ionic groups are nothing but they are certain parts of the enzyme that can be ionized, or it can form a negative or a positive charge within that particular area of enzyme or within that particular group of enzyme. Now, these are the areas that actually react with the substrate when they form the enzyme substrate complex. So these particular areas, since they are ionic in nature, that is why the pH of the solution in which the enzyme is functioning plays a very crucial role, because most enzymes have a very optimum pH at which they show maximum enzyme kinetics or maximum activity. If you can see the image over here, we have seen that when pH is plotted on x-axis and enzyme activity is plotted on y-axis, we see that one enzyme, which is pepsin, shows maximum kinetics at 2 pH, whereas salivary amylase or trypsin shows their activity at maximum activity at 7 pH or maybe around 8.9 or 9 pH. So you can see here that different enzymes have their favorite pH at which they can show maximum activity. This is called as the optimum pH at which the enzyme shows maximum activity. So friends, why this is important to us is because the reagent system or the reagent that is provided in the kit, in that reagent, already the pH is set to optimum pH at which the enzyme will show maximum activity. But if these reagents are not handled properly in the pathology laboratory, then it may change the pH of the reagent. And when the pH of the reagent is changed, then the enzymes, even though they are present in the reagent, they will not show maximum activity. And this will eventually lead to an erroneous result. That is why it is important to maintain the pH of the reaction mixture. Now let us look at the second component that plays a vital role in enzyme activity. The second component is the ionic strength of the buffer. Now, what exactly is the meaning of buffer? Buffer is any solution that resists the change in pH, which means that the buffer solution will not allow the pH of the solution to change. How this is important to us is because in our pathology laboratory, most of the tests are being done in the test tube, and the test tube is not covered. It is exposed to the environment of the laboratory. Now, we all know that in the environment, in the gas, there are various gases which are present, like, for example, carbon dioxide, or maybe oxygen, or all the other gases are present in the environment. These gases also, they come in contact with the reagent or the reaction mixture. And when gas comes in contact with the reaction mixture, it forms certain acids or certain base in the reagent mixture, which tend to change the pH of the reagent. That is why the reagent is made of a particular buffer. So buffer is the actual scientific thing that resists the change in the pH. So how much pH it will resist for the change, that depends on whether the buffer is a weak buffer or a strong buffer. A weak buffer can resist the change of a pH only to a limited extent, whereas a strong buffer can resist the change in pH for a good amount of extent. Now we have seen this particular concept is called as ionic strength of the buffer. If the buffer is of a strong ionic strength, or if the buffer is too much weak ionic strength, in both these conditions, enzymes don't function properly. Therefore, a good reagent is always a balance between the optimum ionic strength of the buffer, at which the enzyme will show maximum activity. So that is why it is important to understand this concept, because these concepts help us to understand how the reagent has to be handled in a pathology laboratory. Now let us look at the third thing that affects the enzyme activity. The third thing that affects the enzyme activity is reagent to sample ratio. Now, why this is important? Friends, whenever we use a diagnostic kit, we open the kit and we always there is a instructions for use or package insert which is available in the kit. When we read the package insert, all the manufacturers they mention that we have to take usually 1 ml of the working solution and we have to add certain amount of sample. Let's say hypothetically we need to add 50 microliter of patient sample in 1 ml of working solution. Now, this ratio of 1 ml and 50 microliter is very, very important. If we change the ratio, again the enzyme kinetics will be affected. To understand this more in detail, let's hypothetically try to understand that we have a motorcycle and we want to find out what is the average that our motorcycle is giving us. Now, till the time where we don't fill enough petrol in the tank, we will not come to know the exact average that the motorcycle is giving. Because if the petrol is too low, then before covering the entire distance, the tank will be empty and we will not come to know the exact average. Therefore, it is sufficient amount of petrol needs to be added in the tank before we do the experiment of calculating the average. This is also similar with enzyme activity. Whenever we have to calculate the activity of enzyme or how much product it is forming per minute or how much substrate it is consuming per minute, then we need to give enough amount of substrate into the reaction system. If the amount of substrate or the concentration of substrate is low, then the enzyme activity will not be achieved at peak. Now, hypothetically, we have seen that when the manufacturer gives in the instructions that for 1 ml of working solution, we need to take 50 microliter of patient sample. In most laboratories, what happens is the reaction is not done in 1 ml. The reaction is done in 500 microliters. So when the working solution is reduced by half, then we have to make sure that even the the sample amount that we are adding in the reaction mixture, that also needs to be half. So when we are taking 500 microliter of reaction mixture, then we need to take 25 microliter of sample so that the ratio is maintained. Here the ratio means for every 1 ml of working solution, 50 microliter of sample needs to be added. So when we disturb this ratio, the speed of enzyme activity will be affected in a way that this concept is called as enzyme or substrate saturation. Now, hypothetically, if we reduce the working solution volume by half, instead of 1 ml, we take 500 microliter working solution. And instead of reducing the sample concentration by half, we take the full sample concentration of 50 microliter in 500 microliter working solution. Then there will be too much amount of substrate in the system. And when there is too much amount of substrate, it actually gives a negative effect on the enzyme activity, and enzyme shows no activity or very minimum reduced activity. This concept is called as substrate saturation. So we have to keep in mind that we cannot add too much amount of substrate and we cannot add too little amount of substrate also. In both these cases, the activity of enzyme will be hampered. Therefore, it is important to maintain the ratio of enzyme to substrate. Now, let us understand the fourth concept, which is the timing of absorbance. Now, friends, let me ask you one question. If we take the enzyme solution in a test tube and we add patient sample in the test tube, immediately from the next second onwards, will the enzyme show maximum kinetics? The answer is no. It does not happen that the moment you add substrate to the reagent, immediately the enzyme achieves maximum kinetics. It does not happen. Usually, it takes some time for the enzyme to achieve maximum kinetics. Now, this particular time required for the enzyme to show maximum kinetics is represented in the form of a graph, and the initial part where the enzyme activity or the enzyme kinetic is not stabilized, that particular part of the graph is called as lag phase. And when the enzyme starts showing the maximum activity or maximum kinetics, then that particular part of the graph is called as log phase. We have to measure the activity of enzyme only in the log phase. We should not measure the activity of enzyme in lag phase, because in lag phase, the reaction has not been stabilized. Therefore, timing of measuring the absorbance is very, very critical. Let us try to understand the fifth factor that plays or affects the enzyme activity. The fifth factor is temperature. We all know that most enzymes give their maximum activity at 37 degree. Why 37 degrees Celsius? Because human body has 37 degrees Celsius internal temperature. So most enzymes are standardized at when they are present in the human body, they function at 37 degrees Celsius. But what happens if we do not maintain the temperature? This, if you play little idea on the graph, if you pay little attention, you will see that the same enzyme, which is like serum aldolase, is the enzyme which is, for an example, used in this particular graph. Now, the serum aldolase activity is different at different temperature. See, the maximum activity is seen at 46 degree, then again at 40 degree, then 36 degree, 30 degree, 60 degree, 22 degree. So whenever there is a change in the temperature, the activity of enzyme also changes. Why this is important to us? Because in IVD industry, whenever we do a clinical chemistry test in a pathology lab, we incubate the test at a particular temperature. Now, if we don't maintain the required temperature, then the enzyme activity will not be at its peak, and we will have a false result. Say, for example, we need to measure the enzyme activity at 37 degrees Celsius, which means that how much product is formed at 37 degrees Celsius per minute. Now, hypothetically, if our flow cell is not maintaining 37 degrees Celsius, will the enzyme show maximum activity? The answer is no. When the enzyme is not showing maximum activity, then obviously the amount of product that was originally supposed to be formed per minute will not be achieved, and less amount of product will be formed per minute. This will lead to wrong result because we will think that actually the enzyme is not functioning properly. But the enzyme is functioning properly, only we have not maintained the temperature. That is why it is critical to maintain the flow cell temperature and do the proper calibration of the equipment and the semi-auto or fully auto analyzer. Friends, I hope this video has been helpful for you. In this video, we discussed that enzyme kinetics are affected by five particular factors: how pH plays a role in enzyme kinetics, how ionic strength of the buffer plays a role in enzyme kinetics, how reagent to sample ratio plays a role in enzyme kinetics, how timing of measuring the absorbance plays a role in enzyme kinetics, and how temperature plays a role in enzyme kinetics. All of this situation is important to us because we face this situation daily in pathology laboratory while performing the clinical chemistry test. We hope this video is helpful for you. If you like this video or if you have any kind of questions or suggestions for us, please write in the comment section. This will help us to improve our future videos. Thank you very much, friends, and I will see you in the next video.